In this video, we will present our company's achievements, measuring and quantifying odors. To begin, let me explain how human senses smell. First, airborne odors travel to the nasal cavity. Then the odor molecules move to the upper part of nasal cavity, reaching the olfactory mucus located on the upper part of nasal cavity, where they are absorbed. The olfactory mucus contains cilia projected from olfactory neurons. Importantly, surface of the cilia contains olfactory receptors, where the odorant molecules bind. Surprisingly, humans have 388 different olfactory receptors. Each receptor responds to various odorant molecules. Responses are of different intensities, weak, strong, or often no response at all. Also, the response curves can differ over time. There are 3 pre-existing odor recording methods. Number 1. Sensory testing. It is a test using sniffing to evaluating odor. It uses human olfactory receptors. However, the disadvantage is it may be subjective. Number 2. Gas odor sensor. It is inexpensive and reliable. However, molecules detected by the devices are limited and do not correspond to human olfactory sensors. Number 3. Gas chromatography. It is a common method and can detect numerous molecules at once. The downside of this method is it also detects non-odor molecules, and analyses become complicated. What does it mean to be quantitative? For example, colors are determined by the ratio of CMYK, cyan, magenta, yellow, and black, as used in color printers. Another example is taste. Taste is assessed by scaling combination of 6 different perceptions. It has been challenging to measure and quantify odor. The challenge is that people's sense of smell is very complicated. First, humans have 388 odor receptors. Second, each receptor recognizes several molecules with a different response curve over time. Lastly, odors are recognized by the combination of several different receptors and responses. By the combination of recognition patterns, people can recognize several 100,000 smells. Our company, Komihakko, made a breakthrough in measuring and quantifying odor. We successfully developed an in vitro system to replicate the human olfactory system and measure and quantify odors. We call it an artificial human nose on a glass slide. Before explaining our technology, let me describe what happens when an olfactory receptor is activated. When an odor molecule binds to an olfactory receptor, the resulting signals are transduced to the calcium channel within the cell. This results in the channel opening and a calcium influx. The calcium signal is transmitted from the olfactory nerves to the brain, which recognizes it as smell. Learning from basic biology, we have developed an olfactory nerve mimetic cells. The mimetic cells express olfactory receptors and calcium sensing protein. The calcium sensor emits fluorescence based on the calcium concentration in the cell. By quantifying fluorescence intensity over time, we are able to quantify any odor molecule. We have now developed a high-throughput system to measure odor systematically. We prepared the glass plate with 20 by 20, 0.5 square millimeters sized microwells. Each well was seeded with about 400 to 500 odor nerve mimetic cells, expressing same receptor. We call this glass plate with 388 wells of each odor receptor as an olfactory array sensor. Here is an example of a recording from our olfactory array sensor. The bar graph represents the intensity of each receptor's response. Each odor molecule activates a different set of receptors at different intensities. In addition, all outputs are recorded from the beginning to the end of odor sensing. Each receptor reacts differently, show different response curves. We call the resulting recordings of intensities over time an odor matrix. In actual recordings, an olfactory array sensor is submerged into the solution with odor molecules. The resulting fluorescent intensities are monitored by CCD camera. The uniqueness and strength of our company. Our technology uses human olfactory receptors, which replicates how people sense odor. The recordings are quantitative and reliable. Gas chromatography is also quantitative, but does not correlate with human odor sensing. Our recordings can be a bridge between gas chromatogram and human sensing. Another advantage of our technology. It is a system with real-time recordings. Why do we need real-time recording? Here are examples of two recordings with a receptor OR2W1. We tested two different odor molecules, 2-heptanone and coumarin. 2-heptanone is referred as having the odor of blue cheese, while coumarin is known as the odor of leaves from cherry blossom tree. X-axis is time and Y-axis is intensity. After applying 2-heptanone, OR2W1 responds rapidly and strongly, then decreases rapidly. On the other hand, the same receptor responds to coumarin slowly and declines slowly. Also, the intensity is relatively mild. A decline in response over time reflects odor adaptation, or so-called nose becoming dumb. These are results from just one receptor. Even so, two odor molecules give different response on both intensity and response speed. Those responses reflect the recognition of different odors. Our olfactory array sensor replicates the human olfactory system. It measures response changes over time. It is a truly one-of-a-kind technology.